I wanted to welcome everybody. I know we've got some time here. We've got a few minutes. Today's broadcast, we're going to cut a little short because I've got an appointment that I got to be leaving on the spot at 4:30. But we've got a pretty much action-packed presentation here on methylation and immunity. And so, what I like to do is I like to see how many people can hear me. Can you all hear me? Raise your hand if you can hear me. Yes, very good. Do you know how to raise your hand? There's like a little hand over there. You can just click on the hand, and it'll actually tell you. You know. So far, I got two hands saying, "Yeah, okay, great." Raise your hand if you can hear me. Wonderful. All right, so we got one more minute, so hang on there, and we'll get a few more people to come on. There's quite a few registrants that have registered. A lot of people register, and then they wind up listening to the recording. But it's nice to be able to have everybody on. And then welcome to ask any questions regarding. I'm going to be taking some of those questions with regards to methylation and immunity. Methylation and immunity is kind of a fascinating topic. And what we're doing in the series is we're taking the first week we took genes and methylations to demonstrate how genes control methylation and how the human genome sets the methylation cycle. And your genome is characterized by your 23,000 plus genes, and every cell of the body regulates at a certain rate determined by the encoding of the genome. And then the second week, we talked about metabolism and methylation and how integrated methylation is in metabolism and how they're interdependent upon each other. And then you're going to see here in immunity how much methylation and immunity are also intertwined. And there's a lot of research out there that demonstrates that. Last week, I actually showed you some charts on the gene chart, the metabolism chart, and also the interface epigenetically on how many pathways in metabolism are actually integrated in with methylation. So, okay, so we are going to begin the process here, getting into our course, and I wanted to welcome everybody. There's quite a few registrants that have actually registered. We are going to be broadcasting this so people can listen to it later. A lot of people, this is a time of day where people are commuting and it's hard for them to get on, but this is the only available space that I had in my schedule. So anyway, it's going to be available for free. And if you have questions, please type your questions in the question section, and then we will actually get into that. All right, so with regards to methylation and immunity, first thing I wanted to show you is that in methylation and immunity, there are approximately 1,839 genes that are involved. So you can actually export this list. You're going to see a lot of HLA, which are the human leukocyte antigens. And you probably remember the HLA antigens that are associated with the gut changes, like people that have a lot of cross-grain reactivity or inflammation in the gut that's provoked by gluten or dairy or any other type of antigen that might be a provocative antigen, will be found on this list of 1,839 genes. Okay, and as you can see, several of them are methyl CpG binding proteins, and the methyl CpG binding proteins are basically islands of methylation, and methylation has a profound influence on antigen processing. So if we actually look at it, and you look at the genome-wide characteristics, this is actually a study that's out there from 2011 that's talking about the DNA methylation dynamics of human stem cells, okay, and how much the methylation cycle actually is involved in keeping the stem cells healthy. And stem cells are generated from your bone marrow, and they are put into circulation to actually assist with immune defense. And a lot of the times, if DNA hypomethylation is, like they said here, progenitor cells right here showed pronounced DNA hypomethylation in monocytes and granulocytes, okay, in this particular study that they've done. And what we want to see is we want to see how methylation is going to actually signal better epigenetic changes. So what you want to do is you want to prevent the hematopoietic progenitor cells from aging prematurely, right? So the methylation cycle is really involved in that whole process of integrity and aging of those stem cells. So stem cells are actually those cells, those root cells that are really important in maintaining healthy immune responses. And we're going to find that as we go along here, that DNA methylation patterns and epigenetic memory is a huge field of study. When we're actually talking about DNA binding and transcription of proteins, when we're talking about transcription of immune-based proteins that are found in the HLA human leukocyte antigen, or what they call the MHC class 1 and 2 complexes, these are really important areas of the immune system where these the transcribed proteins for assisting with epigenetic balance are crucial. Okay, so crucial patterns in DNA binding, which we find that DNA methylation and epigenetic memory and immune responses are very intertwined. So as we go along here, you're going to see those relationships. Some of the antigen processing and presentations of certain genes on certain CG identities. These are actually some of the genes that are tabled here, which are known as the HLA, DPB1 and DRB1. A lot of these immune-related CpG methylation expressions can be found to be an issue in autoimmune responses. So which means that if a person has a lot of autoimmunity, that seems to be characteristically demonstrating in chronic, maybe fatigue-like changes, maybe evidence of Hashimoto's thyroiditis, which is an autoimmune disease, SLE, Sjogren's. These are, non-specific tissue, connective tissue autoimmunity. This is what we're talking about, how methylation is definitely, if we have a problem with methylation with hypo or hypermethylation, or extremes that way, can profoundly affect the processing of these antigens. Okay, and then as we're going along here, this is a really good study here that's talking about the role of gene methylation movement in anti-tumor immune responses. So if you can see, I've shared my screen here, so you should be able to see the screen. Really important. But the most important thing to remember that with regards to immunity is that if we have any kind of methylation defect that's occurring, it can change the landscape of the immune system to not be as keen on detecting tumor cells. And tumor cells will then evade recognition by the immune system and will not be destroyed. And a lot of the things here that we see in relation is that the methylation movement, if it's hypo or hyper, without corrections, can definitely encourage bad epigenetic signals, and you would find more mutations that occur in response to that. This is a very good study. Some of you out there may not be looking at the science of it in detail, but some of you may, and I encourage you. These type of studies here are very, very informative in helping you to understand the DNA hypo and hypermethylation patterns. And you can actually, there's a lot of talk going on right here about DNA sequences and cancer-linked DNA hyper and hypomethylation patterns. Okay, so I would encourage you to look at this study. I will, I have some studies from last week and this week that I can send to your emails if you desire that. You can just let me know and say that you would like to have those articles, and I can actually send them to you. Okay, these are pretty recent studies. They're not old, 2011 on this one here. Now, this is interesting with environmental triggers and autoimmune expressions on methylation and epigenetic dysregulation, or what they call deregulation. So the ones that were studied here are the SLE, rheumatoid arthritis, type 1 diabetes, and multiple sclerosis. But you can see the common autoimmune reactive patterns in several types of disease patterns. And you're going to find that methylation movement and defects in methylation movement will profoundly affect the immune system's ability to correct a complement or recorrect itself after an exposure. So, for instance, if you have a viral exposure or microbe exposure or some type of bacterial exposure or an environmental exposure, the whole idea is to help create a balanced tolerance so that the immune system responds to the exposure, but then will then go back to a normal tolerance so that there are no autoimmune expressive changes. And oftentimes, what you're going to find is that the autoimmune expressive changes are associated with uh, a failure of recorrecting in the methylation cycle. So there's an inner relationship there that I think you need to pay attention to with regards to any type of autoimmune reactivity. And the inflammation patterns are associated with that. So chronic inflammatory diseases, which we're going to get into methylation and inflammation on our the next session, which is really important. So you'll get to see some of the methylation inflammation gene relationships. Very, very keen on looking at that. And then another good study that was actually done in France here. It was a very light study back in 2011. Well, actually 2010, but it came out in 2011. Is talking about the first clue of evidence of autoimmune reactivity in animal studies. But it's also, we also see the same type of patterns with T and B cells in human studies. And so the first clue of inhibition, as it says here, in SLE was the DNA methyltransferase induction. We usually would see DNA methyltransferases being affected, and then that's how you know that the immune system is actually going to have expressed changes in how it responds to any type of exposure or any type of threat. And then if it doesn't recharacterize itself in this methylation process, you're going to find that these DNA methyltransferases are the induced system of creating more ongoing autoimmunity. And down here, it's saying that it's also been shown that DNA hypomethylation of T cells and B cells correlates with reduced DNMT efficacy and histone acetylation in SLE. So when we actually talk about promoter genes as being demethylated, uh, there's a fine balance between the methylation movement of hyper and hypomethylation in the DNA methylation status and the accelerated autoimmune type of symptoms. And so we really need to, you can see the profound effect on T and B cells, which is really a huge relationship there. So I think that with that, you can see the value of being able to look at this and say, how is my immune system affected with my methylation? How does my methylation cycle affect my immune system? And basically, what you can see from some of these studies that I just point out just briefly, which like I said, I can get you some of these studies and you can actually look at it yourself, is that methylation profoundly affects immune response and immune correction. Okay, what comes into play also is the chronic inflammatory response as well. So if you have, continuous inflammation with threat of autoimmunity because of some exposure, for instance, some of these genes that I'm talking about, these mutations that you see on these reports that you're getting back from 23andMe and some of these other conversion reports, is you probably would notice HLA DQA1, HLA DQA2, DRB1. You will actually know HLAs in general. Some of the ones that are you're going to notice the other antibody-associated changes on some of these SNPs. But I'm mentioning a few of those. But some of the inflammatory ones too, like TACR1 and the NR1I2, which is another one, the CETP, the CRP. These are all inflammatory-based gene SNP variants, but they're very much intertwined with the immune HLA DQA ones that we actually see, DQA1, DRB1, some of those that you see. You can even see the ones that are associated with ANA, anti-double-stranded DNA, These are all antibody associated. And some of the other immune ones are known as the FCGR2A, the FCER1A. These are all antibody encoding genes that are also related to autoimmune responses. So there are several mutations that we can see on these reports that can demonstrate these patterns. And I really highly encourage you if you do have your report, to find someone that actually can assist you with identifying immune sensitivities and inflammation sensitivities, because it can kind of give you a good clue as to what we could be looking for if you have a chronic standing problem that you just can't seem to shake. And then there's some corrections that can occur if you can at least identify what gene sensitivity may be there. Okay, so now I'm going to be taking some questions here. Does anybody have a question regarding methylation and immunity? Just feel free to type it into the question box, and I'd be happy to answer that. We have one person that mentioned that they're interested in receiving some of the studies. I will actually pool the ones that I got from last week and this week. Make sure that you include your email addresses, and I can send them to you. Okay. Now, the most important thing to remember is that in the DNA methylation status, DNA methylation profoundly influences a renewing effect on both metabolism that we talked about last week and immunity. So when we're actually wanting to balance out methylation, the first thing we have to do is find out where the burden where the biggest burden on methylation exists. So if there's a high burden on methylation in other systems, like for instance, if a person has chronic cardiovascular related inflammatory disease that's occurring, they can develop autoimmune reactivity against their own vessels, which can create more cyclical inflammation. And in that particular case, methylation is going to be probably most likely to be found to be hypomethylated. And then we're going to find that unless we really tone down the negative effect on the vascular system by eliminating the inflammatory cycle by, dietary changes, lifestyle changes, bionutritional movement, exercise, all of these different things have profound epigenetic influences on that. And we really got to pay attention to that. Okay. So that's really essential to know that methylation is you got to find out where the assault on the methylation is occurring. Okay, so that's very important for us to know. I would suggest anybody that wants these studies to please send an email to info@healthcoach7.com, info@healthcoach7, the number 7.com, because when you put your email addresses on this, the thing is that I may not remember those emails, and they may not come out on the screen here appropriately. So please, info@healthcoach7, the number 7.com. All right. Another question that's out there is, if I have a high SED rate, how do I know that I'm having some kind of an autoimmune problem? Well, a SED rate is not really a good way of trying to determine whether an autoimmune problem exists, because a SED rate is kind of like your first stopping point on the blood. It is actually basically a phase reactant response. You really need to be doing a couple of different things if you want to know if you got autoimmunity happening. One of them might be looking at an HSCRP, which is a high sensitive C-reactive protein. Another one is an ANA. Another one is an anti-double stranded DNA. Another antibody responses are antibodies against thyroid gland, like thyroid stimulating immunoglobulin or anti-TPO or an anti-thyroglobulin. You may want to have your HLAs assessed, like HLA DQA1 and 2, DRB1, some of these other ones that are involved with connective tissue disturbances and ongoing autoimmunity. Now, sometimes when you have this persistent autoimmune reactivity, some of these things can actually cross the blood-brain barrier, and you want to be looking at what the antigen-antibody ratio is so that you can actually assist the body in calming down the reaction, okay? Because if you can't calm the reaction down, and I'm not just talking about putting a person on steroids to do it, because that's immunosuppressive, and then you'll have a whole lot of other problems to deal with. We want to be able to take the first steps of elimination and changing lifestyle, changing diet, changing any antigenic provocation, and doing our best to really spice up the immune system with bionutrients that it likes to correct this pattern before you start running to immunosuppressive agents, which I firmly don't believe solve a problem at all, or even some of these more sophisticated drugs that are out there that are designed for autoimmunity that actually may cause some other type of cancers to exist. So I would say think about those type of testings. That's a basic. There are other ones out there like Cirex Labs actually does a whole array of looking at neuroimmune autoimmune agents as well as systemic autoimmune agents that might be provocative. And the other thing is you might have ongoing assaults that include like heavy metal changes and maybe shortfalls in the metabolic process that we talked about last week. So there might be some metabolic issues that are creating the imbalance. Okay, so that's a very good question. Are there any other questions that you have out there? Can you still hear me? Everybody raise your hand if you can hear me. I got one person that raised their hand. Raise your hand if you can hear me. That's okay. We have a few more people that are raising your hand. Okay. So anyway, I'm really feel privileged to be able to provide this information to you, and like I said, I will provide these studies for you. Getting in more depth within a half an hour is hard, but I just wanted to demonstrate to you the interrelationship of methylation and immunity. And remember this, that if you are a good antigen processor, you're going to have less autoreactive issues. If you are not a good antigen processor and you're having maybe some susceptibilities there, you are going to have more challenges in trying to auto recorrect. And if you have any exposures like viral exposures like cytomegalovirus, Epstein-Barr virus, HHV6 virus, and some of these other things, that's going to profoundly affect your body's ability to how to respond and recorrect itself. All right, very essential to pay attention to those things. Yeah, another question out there is a very good question is, can you talk more about mold exposure and methylation? Now, the mold is such a very complicated problem because the immune system, what winds up happening is it's actually a cross between a low to moderate grade inflammatory movement that goes on for a long period of time, because most of the time people don't really know that they've been exposed to mold over a chronic period. So like in other words, some people that I've actually worked with have been sick for years until we discovered that we have mold exposure. Now, the methylation cycle in mold exposure is very much the same as what you would see in any type of autoimmune reactivity. And a lot of the times you will see crossovers in hypo and hypermethylation extremes because the provocation and complement with mold is huge. And the thing about it is that mold is not easy to get rid of. Okay, look at mold and candida. Okay, if you look at candida species, candida produces spores. A lot of these other molds produce spores. But the thing about it is that some toxic molds actually produce what they call toxins that actually circulate through the blood and are very difficult to get out from the liver. So the P450 system becomes bogged down, and it cannot deal with the heavy toxic overload that's produced by the toxins that the mold produces. So the methylation cycle for sure will be greatly affected by continuous mold exposure. And like I said, the problem here is that mold, most people do not know that they're exposed to it, and they can be exposed for long, long time, many, many years before they're actually worked up appropriately. And there are panels that can be done to test for specific molds of regions, as well as general mold patterns for looking at IgG and IgE reactivity. And there's even more sensitive tests out there that can actually pick up on looking at what they call probe mold. Uh, um, they're actually called mold probes, which actually the mold probes actually are better because you can actually see if there's any trace remnant of genetic association. And more labs are actually getting into more PCR uh, effects in being able to evaluate mold and bacteria more effectively. So, and Genova Diagnostics is one of those that actually does right now a GI effects on PCR. There are some limitations, however, but we're going to find a lot more proficiency. So there's a lot of labs right now that do this mold detection. And yes, methylation is definitely affected big time with mold exposure. So, yeah. All right. That I think, let's see, we've got a couple more questions here. Traditional labs usually don't run some of these tests. They might run some like the viral testing and the inflammation testing and some of the traditional auto antibodies. Yes, so, they do run, but sometimes what happens is the doctors that are ordering these tests are limited by diagnostic criteria. So, like in other words, they have to match the diagnosis, and the codes actually have to match in order for the insurance to say that they would cover it. So there's like a bunch of hoops that have to be jumped in order for insurances to, cover. And then, as I mentioned earlier, another person asked, is everyone with amalgam type fillings, could they be affected with affect methylation and immunity? Yes. So, because of the heavy metal toxicity associated, you have a chronic immune activation that occurs with heavy metals, including mercury, nickel, titanium, lead. And there's a lot of that stuff going on in prosthetics as well as implants in the mouth. So, it's not just mercury that's the problem or amalgam fillings, it's the others, prosthetics too, that can be a problem for some people. And then also the prosthetic and the high risk of biofilm development with foreign material being used. I mean, a lot of times you can't get around that, but you got to know if whether or not biofilm is a problem with prosthetic replacements of any type, whether it be implants in the mouth or surgical replacements like hip, knee, et cetera, et cetera. So I would say check with your insurance company for coverages always, and you can always run a list by them of what these tests are before you actually have them run and find you have a big bill. And then making sure that your doctor is actually cooperating with you. And then for like mold testing and things like that, for the person that brought up that really great question about mold, um, you want to know what your methylation cycle is, where it's at in the sensitivity. So 23andMe raw data that you have, you may want to be looking at getting a genetic sensitivity susceptibility evaluation to tell you more about whether or not you have susceptibility. And then you may want to get yourself a good mold exposure panel going and looking at some of the associated gene SNP susceptibilities for mold. And there are a lot. You can actually look up mold and mold and immune response. I mean, if you look up in gene cards can actually give you topics that you can actually input into the search window, and you can discover maybe what some associated genes might be in mold relationships and the immunity. So there are a lot of genes that are actually respond to that, and a lot of them are interleukin ILs, 1, 2s, 6s, CD14s. There's different receptors that actually respond to mold. So you're going to see a lot of immune gene controls that respond to mold, bacteria, and viruses. Anyway, I'm running out of time. It was great to see everybody today. We had a pretty good showing, and I want to just say thank you for attending. Please do attend next week. Share with your friends, because these are free seminars, education to give you at least a sense of maybe what you're going to be picking up and saying, oh, you know what, I never really thought about that. And that's something I wish I could be doing, or I should be looking at more detail. If you have chronic problems going on, if you've got concerns about heavy metals, concerns about what to do with your teeth and your gum health, like with root canals and all these other things, please, be aware that there are better ways of working it up. And a great starting place would just be to show you what your gene sensitivity and susceptibilities are to kind of get a clue of how to approach it. Very, very important at this point to consider that. And I look forward to to next week's broadcast, and we're going to be talking about, I believe it's inflammation, so it's going to be the methylation and inflammation effects. And I think you're going to find that that's going to be very, very interesting, in how the crossover in how it's related to the immune system. So I will get these studies out to you. Please, email info@healthcoach7.com. This will be available on the recording. You can listen to it again, and you can share it with your friends. And then we'll actually see you next week. Thank you, everybody, for attending. It's really great to have this opportunity in working together to understand genetics and epigenetics and functional health. Until next week, we'll talk again. Bye-bye.